Okay, I'd like to start us off by acknowledging that this virtual presentation is taking place throughout the unceded territory of California, home to nearly 200 tribal nations. In particular, I am speaking to you from Patwin, Miwok, and Nisenan ancestral lands in Sacramento. We acknowledge and honor the original inhabitants of our various regions. We encourage you to learn which tribal nations are indigenous to the area where you are now living. One excellent source of information is Native Lands, and we've dropped the link into the chat for you to explore later. I also want to acknowledge that we are coming into this meeting today upon the news of losing another Black life, Dante Wright, to police brutality. This additional loss to the many we've experienced within the last year weighs heavily on us today and reminds us that our justice work continues to be urgently vital for us all. And with that, I'd like to introduce our speaker. I first learned myself about CalEDNA, about some of the amazing work that the team has really done in the last few years in a presentation with the Center for Community and Citizen Science here at Davis, and they were presenting about how the data collected about environmental DNA has really filled in a lot of gaps about what we're seeing out in nature that iNaturalist doesn't necessarily capture. So it was so cool to learn about that. So I'm really excited for our speaker, Dr. Maura Palacios Mejia, to come and talk to us today. She's a certified California naturalist herself from Domingos Rancho Adobe Museum in Rancho Dominguez, and a postdoctoral scholar with the Howard Hughes Medical Institute at UCLA. Her research focuses on applying environmental DNA, or eDNA, to address issues in conservation management and urban planning. Her projects use eDNA to assess species assemblages in the Mojave Desert Springs, a threatened ecosystem, and to explore microbial community changes in relation to hazardous materials at Brownfield sites throughout Southern California for potential bioremediation practices. Dr. Palacios Mejia also uses eDNA as a tool for research and education and outreach. She is a recipient of the Sloan Scholar Mentoring Network Career Development Grant to carry out a new project in collaboration with the Nature Conservancy titled Establishing a Baseline of Biodiversity and Ecology of Jalama Creek in the Jack and Laura Dangermond Preserve using environmental DNA to assess stream restoration success. So Dr. Palacios Mejia, take it away.
Okay, thank you so much. Welcome everyone. It's a pleasure to be here. I'm really excited to share with you a little bit about the research that we've been carrying out as a part of the CalEDNA program, and also some potential ways that you can get involved with our group in the near future. So I will start sharing screen. Right. And the title of my talk is the CalEDNA program, Transforming How We View Community Science. So just a little bit about myself. I'm currently a postdoctoral scholar at UCLA in the Wayne Lab. And the Wayne Lab specializes in using environmental DNA, but also focused on using genomic tools to be able to conserve and protect endangered species. I completed the California Naturalist program back in 2018 with a cohort of several amazing people. I have been currently working on using environmental DNA on different types of projects, and I'll be explaining a little bit about them as we go through the talk today. So you may be wondering what environmental DNA really is. So this is just DNA that's found in the environment as organisms interact with it. They will leave traces of DNA behind, and that could be in the form of pollen, blood, urine. In the case of like bacteria, it'll be whole cells. And then we can go ahead and collect that from water samples or sediment samples, and even air samples. If we just filtered some air in a room, we would collect it on a filter and we would be able to get some DNA from that. It's been a very useful tool in detecting the biodiversity found in different types of ecosystems, which will be exemplified as we go through the talk today. And so it's a very new tool that has only been around for maybe about 10, 15 years, and only now are we honing in how to properly apply it. And so I'm going to walk you through just some of the ways that I've been using environmental DNA in my research projects. So we recently did a pilot study where we tried to compare 4 different Mojave Desert Springs that can be seen here by the A, B, C, and D. And here are some images representing what that looks like. So what we did is we collected water samples and sediment samples, and then we brought those samples back into the laboratory. And then we tried to compare what we found with environmental DNA to camera trap data, where we actually had poor agreement between what we detected with eDNA and what the camera traps picked up. We then also compared to botanical assessments that were done in the field. And these also had some low to moderate agreement. And I guess I should say that for the camera trap data picked up much more data or organisms than we could with environmental DNA. However, eDNA picked up 12 species that were not detected by the camera trap data. And then for the botanical assessments, we had low to moderate agreement, where eDNA tended to find way more species than were done by the botanical assessments. We also found high agreement with birds. So birds are very popular, very obvious, and there's a lot of documentation that goes into tracking birds. And so there was high agreement between what we found with eDNA and what had been observed on site. And for vertebrates, it was pretty high. We had an 82 to 86% range. There's some links here to some of this research that has been published as a result of this study. And this was in collaboration with the Nature Conservancy. I'm also doing a project focused on brownfield sites. So these are sites that have been abandoned because of the potential presence of hazardous substances on site. And this project is in collaboration with the Nature Conservancy and also the city of Los Angeles. So normally the parcel will be abandoned, and then we will go in and collect core samples to a specific depth so that we can trace to see how deep into the soil some of these substances may be. We then subsample the cores and take samples and then bring those back into the laboratory, and we amplify for any microbes. So this will include fungi, bacteria, and eukaryotes. And why we're doing this, the goals for this is to be able to identify native microbes that have the potential of metabolizing some of these hazardous wastes and then using those to inoculate it on site for a natural form of remediation while maintaining the native soils and the native bacteria and microbial communities. So we also recently received some funding from the Sloan Foundation to be able to carry out on a new project that we recently started. And this is in Jalama Creek, which is in the Dangermond Preserve near to Santa Barbara. And this is a historical site where we had steelhead trout runs. However, there has been a lot of modifications of the stream to reduce flooding. And so there's these restoration efforts that that they want to happen in the preserve. And so what we're doing is we're using eDNA to be able to establish, one, what is the community that is found there? Two, do we actually pick up any traces of steelhead trout presence in these creeks? And then there's also the sensitivity of finding endangered species that are also present and on site. So based on these results, we will then be able to create an improved management plan into the particular locations and how we approach the restoration of it. And the plan is also to continue our eDNA sampling and to be able to monitor progress over time. And so we know that when restoration takes place, the results aren't immediate and it can take several years. However, some indicators that are able to tell us whether the practices of remediation are good can also include microbes. And so we can use those as indicators to be able to tell that that particular soil is coming back with the community that we're expecting. And this year, we just got recently funded for a new project focused on marine ecosystems, particularly kelp forests and seagrass meadows. And we'd like to, so this is a collaborative interdisciplinary project called Kelp Magic, and it encompasses 7 other researchers at UCLA with different specialties. So we have someone that focuses on genetics, we have another person that focuses on law, we have someone else that focuses on urban planning, we have someone that focuses on spatial science. And so we're a very diverse team. However, we would like to use a community building model to be able to create a more sustainable Los Angeles. And this is part of the Sustainable LA program at UCLA. And if you'd like to get involved, there's a link here where you can connect with us so that we can include you to participate in conversations and workshops and webinars that we'll be having starting in this summer. And you can also reach out to me if you'd like more information. But the goals of this project are to integrate culture, economy, and technology to be able to use our natural resources in the ocean to be able to better prepare for a more sustainable future in Los Angeles. And how does that fit into the CALeDNA program? All of these projects have in one way or another included community scientists. And so what is the CALeDNA program? This is a statewide program that has started about 7 years ago through funding through the Genomics Center at UCLA. And as of now, how the program has worked is that you register, you sign up for BioBlitzes, you collect samples, and then we're able to process those samples in the lab. We make all that data available to the general public, and you're able to then explore the results. We have over 1,500 sites sampled all throughout California. That has included a wide diversity of organisms that range from plants all the way down to tardigrades. And we are trying to continually be inclusive and include all community members. If you're interested in learning more, you can visit our website here, and you can also sign up for the newsletter. And then that is a way that we can communicate with you of any future events that we'll be hosting. And I'll talk about some events that we've had so far. So we also started 2 years ago this Environmental DNA for Science Investigation and Education program, or the EASY program. And we've embedded the CALeDNA model into the classroom. And so we have 2 courses. One is a field-based class where we take students to different locations all throughout California, particularly Southern California, because it's the closest. And then we also have a fall course that's more hands-on and focuses more on data analysis and understanding how the process of environmental DNA works. We also have a summer internship program that has been going on for the past 3 years, where students carry out their own independent research based on environmental DNA. And here are just some examples. So we had a student focus on a fish die-off of 2018 in the Malibu Lagoon. We have another student who worked on biodiversity assessments in the Mojave Desert Springs. And then we had another student focus on spatial and temporal variation of environmental DNA in kelp forests. And so you can see that this method is really great at engaging people wherever, at all different levels. And so that's the great thing about environmental DNA. So I wanted to just focus a little bit more about the class because it's like I said, it kind of uses the framework of the CALeDNA program. So we students participate in collecting observations using iNaturalist. They also participate in a DNA barcoding project where they collect specimens and then extract the DNA and then are able to identify based on the genetic sequences of them. We integrate environmental DNA sampling, and they also do a field notebook. So in the field, students have done, this is for the, they've done both eDNA sampling, but also insect sampling for our more recent project on the on the Los Angeles River. So students collected insects from malaise traps, sweep nets, and also D-nets in the actual stream where they collected macroinvertebrates. They took photos and then each student was assigned whatever insects they collected, and then they were able to process those for the DNA barcoding part of it. They also collected at Sage Hill. For this case, we actually set up pitfall traps. They were set up the night before, and then malaise traps were also set the night before. And then the next day we were able to retrieve them, and then they were able to get that access to that data. So we're trying to expand on this DNA barcoding approach. So this is another way that if you're interested in participating and getting involved, we're going to have several weekends where we want to carry out some events at Stunt Ranch. We're also trying to integrate the botanical garden at UCLA and different green spaces to bring in different community members, obviously under COVID restrictions, so the number of participants will be limited. But if you're interested in participating, there's also be a link shared out to you. And we're going to be starting at the end of this month and all through May. If you're interested in participating and you're local here in Southern California, we welcome you to come join us out there. And this is a project that's in collaboration with Cold Spring Harbor Laboratory. They have developed methods to be able to do this in your house. And so we're trying to incorporate this as a method to do in the field. We provide you with all of the equipment that you need. You will collect insects and plants, and you will do your extractions, in the field. So this is another project that we recently started. This is at the in collaboration with Palos Verdes Peninsula Land Conservancy. We actually just had an event this weekend on Saturday where we had a team of, I believe, like 20 participants, volunteers, and different team leaders. And we are trying to do a project where we're trying to figure out different restoration indices. So they have several locations that differ in terms of whether they've been restored in the past, whether they've been untouched and are just the natural state. And participants came out, collected some samples, and then we're hoping to be able to turn that around in about 3 months to be able to provide those results back, published on the CALeDNA website, but also to the team. And based on this information, we'll be able to understand the process of, or the success of restoration in these areas. We've also launched a brand new project on the Los Angeles River, which is called Protect Our River. This was launched about last year, and the goals of this project are to just increase community around the Los Angeles River, and to develop some biomonitoring around it. So there will be some links for you to be able to explore data, submit artwork, and there's an iNaturalist longstanding page of observations that definitely helps us to be able to compare the environmental DNA results that we get to the observations that we would expect. And another project that will take place next week is we have an event in Pacoima, in collaboration with Pacoima Beautiful and Nature for All at their edible garden, their sidewalk edible garden. We have been holding workshops last month focused around different ways to advocate for the jardín, to be able to communicate effectively, also to be able to express yourself via art, where participants were provided with different kits to make signs for each of the plants at the jardín. We also explored the idea of alternative irrigation systems like ollas, which is a method that is like 4,000 years old, where you put these like terracotta planters in the ground, and then you fill them with water, and then you don't have to continuously go water them. And what the objective of this is going to be, the celebration. So we had about 20 community members participate in this. This is going to be open to the general public. So feel free to come out. We're going to be putting out our signs, putting the oyas in the ground, giving away seeds, tomato plants, and just celebrating right before Earth Day, the accomplishments of the community. We've also been able to collect some samples for eDNA, where community members collected samples from their backyards, where we were trying to compare microbial communities in relationship to fruiting trees. And those results are actually interesting because our expectations were that the communities would be fairly similar if they were from the same fruiting tree, and we actually found that those patterns didn't hold. And so we may want to explore some additional environmental variables and as a result of that. And with that, I just want to thank, there's all of this work is a lot of people involved. I can't name everyone, but these are a lot of the collaborators that have contributed in different ways to be able to carry out a lot of these projects. And so if you're interested in reaching out or interested in getting involved, feel free to email me or get involved through the newsletter. But that is all my presentation for today. And I will take any questions that you may have. Thank you.
All right, if we have any questions for our speaker, go ahead and put them into the chat. And looks like we have one asking about how we get so many different stakeholder groups to cooperate.
You know, there's, with the LA River Project, it's been particularly interesting because we see, you know, different groups having their goals in mind with what eDNA can do for them. However, like, there's a lot of interest in protecting the river, in restoring the river. And so ultimately the goals are very similar. And I think the beautiful part about environmental DNA and just science as a whole is that there's no politics behind it. You know, there's no, you know, driving factor. It's a very inclusive form to just get out in nature and learn something new. And so I think the great thing about it is, despite each group having their own particular interest with eDNA, as a collective, we just want to improve our environment for long-term generations. And I think that that factor, that underlying factor, is what brings a lot of people together, despite, you know, differences.
Another question here is, do you need organizations to provide the environment for a project, or do you want the organization to propose a project?
I think it can work either way. A lot of the projects that we've talked about have come about because an organization has reached out to us and they're wanting to use environmental DNA with their volunteers, for example. And then we've worked together to design, you know, what would be a great question given the area that we're working in. In other cases, you know, there's areas that need to be explored. And so they're like, help, we would love to cover all of this area. How can we use eDNA? And so I think it can work both ways. And I think, yeah, so whatever interests you may have, I think we work with you to make it very personal, depending on what your needs may be.
We had someone wondering if there was anything offered for Eastern LA County.
So right now, because of COVID, we've kind of limited our bandwidth of different projects that we can carry out. If you have a particular project in mind, just feel free to reach out to us. We're always open to new collaborations. And as of the moment, I can't think of a project that we do have in Eastern LA County. But if you are interested, feel free to reach out to us and we would love to work with you.
All right, well, I'm not seeing any more questions in the chat. Oh, here we go. Is there a confidence threshold you might have for mistakes that are made by community scientists, similar to INAT kind of has the different levels of confirmation there?
Yeah, so with environmental DNA, it's a really great tool in two ways. One is that when you actually go and collect the samples, we train you from beginning to end. You do a training, we train you on the spot as well, and you really can't go wrong with collecting a soil sample or collecting a water sample. I think once we actually get the results, however, there are some discrepancies that can occur with our results. So for example, some of our students were, looking through the website and they found dinosaur. Or another thing is like, there's been several people to reach out to us about like cryptids. They're like, have you found any Bigfoot DNA? And so, there's, yeah. So what I'm saying is, I think it's not necessarily the errors that you can make in the field per se, but I think it's just sometimes with the results and how we manage the results and putting those online and how those can be kind of a tricky thing to navigate. I hope that answers your question.
Yeah, I like this question here. Tell us a little bit about how the DNA data is stored and where you share it, who you share it with.
Yeah, absolutely. So all of our results are made publicly available on the CALeDNA website. And I mean, I don't know if you'd like for me to do a quick demo to show you how to do that, if that would be helpful.
Yeah, we have a little bit of time between presentations.
Yeah, I'll try to keep it brief. Sorry. Let me share. Sorry about all the tabs, but this is what our website looks like. So you can explore data here. We have different particular projects. It talks to you about how the samples were analyzed. So if we just click research projects, here are all of the locations that we have explored so far. And maybe you have you want to focus on the Los Angeles ports. You can go click that. This will tell you a little bit about what the project was about, what were the goals of it. And then it creates a little map that shows you all of the locations that were sampled. And then you can just go in there and actually click a particular sample that you're interested in exploring. And then that will provide you all of the information that we obtained. So here we have all the animals that were detected, bacteria, fungi, plantae, and all of that. And then you can explore that a little further. So I hope that helps. I'll stop sharing.
That'll be fun to look through for people, I think.
Yeah.
Let's see, we have, can you elaborate on the Vista Hermosa Park restoration and how eDNA is used to help guide policy?
Oh yeah, sorry. This is just an example. So of how a location has been restored from a site that was a brownfield in the past and how it can be changed to a park and a community location. A lot of brownfields have that goal in mind, either to be converted into housing, but the majority of them, the goals are to convert them into some kind of park or some community type setting. And so when we get the information from the brownfield type data, what we're trying to do is we're trying to figure out two things. One, can we use this method as a lower cost alternative to what's currently being done for remediation? So normally what gets done is all of the soil gets taken out and removed and then replaced. And so we'd like to kind of change that model by trying to use eDNA as an indicator and then trying to use eDNA to identify microbes to be able to remediate on site. So hopefully that answered the question.
Sarah, yeah, definitely.
One question, Dr. Palacios. These are really great examples and projects. As a California naturalist yourself, I'm wondering if you have some other examples of how local communities have used CaleDNA to address kind of locally identified or driven issues or problems.
Yeah, I think at the moment, the majority of our projects have been focused on locations that we tend to have permit for. And so those would be, for example, locations that are owned by the UC system, which are like the reserve systems. And it's a little bit more difficult to try to address community-driven goals. And I think that that's what we're trying to do with the kelp forest project and the, the seagrass bed, the marine kind of project, is no longer are we setting like what the project should be, but what is the need and what are the interests and how can we together start to develop solutions for issues that as a community as a whole we have.
Thank you.
Yeah, it definitely looks like people in the chat are wondering if there are projects or collection sites near them. So if you could just share how people can find that.
Oh yeah, so if you sign up for our newsletter, we continuously send out posts. So we just had our first in-person event was this weekend because as you know, like we've had the COVID restrictions for quite a while. So we haven't had any in-person events. So we had our first one this weekend and we're hoping to increase those. And I shared a little bit about the opportunities to go and sample at Stunt Ranch. Not only will you be doing DNA barcoding, but we'll also be collecting eDNA samples. So we're trying to merge the two and try to, I guess, maximize the experience for our participants out in the field. But if you sign up for the newsletter, they will send you information there about upcoming events, webinars, anything that's going on with CALeDNA, you will be able to find out that way. And if you have any particular projects in mind, you can also reach out to me and I can put you in contact with our lead for the CALeDNA program. Any other questions?
I think that about wraps it up for this portion of the presentation. So thank you so much. Maybe if you want to stick around and browse the chat to see if there might be anything you can answer directly, and then we'll move on to part 2.
Absolutely. Thank you so much. All right. Thank you.
So just 1 second while I get the setup going. Oops, my computer decided to freeze here for a second. So while I'm trying to open the presentation, I would like to talk to you today about how a perfect segue here for CalEDNA and how it fits into the greater participatory science projects that we are doing within the California Naturalist program. Let's see. Sorry, my presentation is stuck. So while we do that, actually, I would like to share with you what we have just created. Which is available on our website. It is, the link is in the chat right now. So this is our new page on the California Naturalist website. So you can find it here. And by going down to Community Science for Naturalists, this will take you to our map. So you can actually explore all the different projects that our classes are doing and kind of get a sense of the scope. So all the different organizations, you can go through them and see what our classes are up to. Here we go. My presentation is back. Here we go. And so what I'd like to talk to you about is how today there's a lot of research that's being done within the UC, and part of the mission of UC Agriculture and Natural Resources, where the California Naturalist program is housed, is to disseminate the UC research to Californians. And increasingly, the dissemination process is actually involving volunteers and Californians and California naturalists in collecting data and being involved in this process. For a long time, different organizations on a more grassroots level have been involving community members in collecting data. And increasingly, the UC is wanting to involve people so that we can all learn from each other and also really expand our possibilities in being able to sort of get more data out there. And here are these are a few examples of what's going on within UC and UC A&R. There's many, many more that are just being born. And the California Naturalist program is really a leader in this endeavor because we have a unique position where we are close to the UC and work with researchers and scientists on a variety of different topics. And then we also have this connection with Californians and everyday sort of people who are able to participate in helping get these data on a wide scale. So with our experience within the program, we really want to show people that we're a leader, our program is a leader, and we want to really kind of get this out there. And this leadership experience comes from our program itself. So we have kind of two ways that participatory science is involved, both at the curriculum level, where within the class itself, naturalists take the data or they get involved in designing the study. All different sort of scopes of involvement with a project happens in class. And then we also have students after they graduate go and volunteer. So I'll talk a little bit about both those different prongs of our involvement there. So each class adopts a participatory science project. As you all probably know, being naturalists here on the call today. And so the class adopts a project and we kind of have put these different projects into these different categories. So within the class, you might take some data on phenology using Nature's Notebook, or you might learn how to take some water quality monitoring data and use some different tools there. And this really helps kind of get a better understanding of how science really works and feel close to these results and kind of feel ownership for what it is that they're studying. And now with Climate Stewards as well, we have some different sort of topics that our projects fall under with some overlap. So increasingly, as we have more courses, we'll see different projects arise from these different topic areas. And of course, we have CalEDNA and all these other different projects that fall outside of the defined categories here. And you can explore, again, what those are on our new tool here on our website, which is really, really neat. So check it out. And so that second part of what the California Naturalist program does and its experiences is that we have our graduates go on and participate in projects that are really a big chunk of our volunteer hours. So it's the second most used category of our volunteer hours. And that ends up being, in the next slide here, almost 50,000 hours reported for the entire program, the lifetime of the program, I should say. So here you can see increasingly every year more and more hours are added, and that means more and more people are volunteering to contribute to these projects. So even last year, you might expect there to be a dip because of volunteer opportunities kind of were put on hold, we have at least a plateau here, which is great to see that. We still have a lot of volunteers going out there, either through their computers or through their backyards to be able to contribute to a project. And that's almost, let's see, if we can look here, that is almost over 300 people a year who are contributing hours and recording them. Maybe there's some people out there who are contributing but not recording their hours. That's definitely true. But all of this is huge impact. And I'm actually going to show you a little bit about one particular area, topic area that naturalists are contributing to. And that is iNaturalist. So if you're a fan of iNaturalist, go ahead and give us the clapping reaction there in your Zoom toolbar, because we're big fans of iNaturalist and the California Naturalist program. And I really like this graphic. It's very interesting to look at because we have now our project, our iNaturalist project for the program that we encourage certified naturalists to join once they're graduated. So I think Brooke will go ahead and put that link in the chat. It's really easy to join. Just make sure you're logged into your iNaturalist account, and then in the upper right hand corner, you'll see a join button. Click that and then you're in. So the almost about 250 people who are part of our project right now, which is only a small fraction of our 4,000 certified naturalists now, are able to contribute these observations kind of in this general spread. So it almost mirrors kind of where we see our partner organizations that offer the course. So at least visually, where you're seeing a California Naturalist course, we're seeing a lot of naturalists continue to record observations to biodiversity science. And I want to show you a particular moment here. These are all the observations in that project that were recorded in 2020. And if you'll notice here, April 24th through 27th, that month is when we had the most iNaturalist observations recorded in the project. And that is also when we had the City Nature Challenge. So right now we have almost 50,000 observations from just those 250 people in the project alone. And the City Nature Challenge is really offering an opportunity for people to really get involved in recording more observations. And the more people that join that project, the more we're able to see the impact of our naturalists to that, the specific iNaturalist project. So I want to show you here from last year as well. So of the 93,000 observations made in California, California naturalists in the top 20 observers for each city contributed 10%. So I went through each of our, 7 cities in California that participated last year. And I just took the top 20, only the top 20, because if I went through everybody, it would be a lot of data crunching. And so all those observations totaled from the 20 observers from these California naturalists within the top 20 observers for their city. Just those alone added up to 10%, and on average, those contributed 14% of the observations to their city's totals, which is amazing. It's only the top 20 people. I can only imagine, after the top 20, all the naturalists contributing to their cities. It's got to be so much more than that. So that is a huge contribution to science. And you can see here, this is a graph as well that shows this exact sort of phenomenon. Even within just the 20 observers of the top for each city, the amount that they're contributing is really astonishing, because they're, especially for the more established cities like LA County and San Francisco Bay Area that have a lot of people contributing observations to the City Nature Challenge, we still have a decent percentage showing in just the top 20 again. So it's amazing to see how many people are out there. So we really want to encourage you once again to get involved in the City Nature Challenge for this year, so that we can really see our naturalists out there, not only enjoying nature, but contributing to this database of biodiversity science. And the same cities that were in the challenge last year are participating again this year. So check those out. You can just Google that in the, for which cities are participating near you and find the project for that one. And we're really excited to see what you all observe, both in our iNaturalist project itself, but also we want to see how you are getting out there. And if you use the hashtag CalNat share, we'll share some of your photos of you in action. Of course, make sure you're masked up and with your pod and staying safe. But we really want to see you all out there, and it's super fun. I will share the project right now for you all to see, because it's super fun to watch in real time how people are contributing and commenting and helping each other, especially right now. As we're kind of staying apart during COVID times, it's really fun to see all these observations from people who have been a part of the program. So with that, I will wrap it up and say thank you to our speaker, and I'll answer any questions about this part of the presentation that I have as well. So thank you. Okay, so I have a question on iNaturalist. If we sign up for the project, how all the observations automatically show up? Yep. So if you look at the project requirements, you have to join the project, so project members only. And anything that you observe within California and is research grade or needs ID will automatically show up. So I bet if I refresh the page that I'll see more people have joined. I like that we're coordinating some people to participate on here. It's great. Is the City Nature Challenge different from this project? Yes, it is. So the City Nature Challenge is organized by LA County Museum of Natural History and the California Academy of Sciences. And this project here is just our program, and it's throughout time as well. So the City Nature Challenge is only for the weekend of April 30th to May 3rd. This is for all time. Let's see. Can you contribute to more than one project for a single observation? Yeah, so anything that you observe, if you're joined in this project, then it will count towards this project. And if you happen to be observing during the City Nature Challenge, it will also count towards the City Nature Challenge as well. And you can really see that. I can actually go to one of my observations, and if I can find one here, you'll see that it gives you a, let's see, here's one. It's easier to see on the desktop version. So if you're on your phone, you can't really see these specific sort of side things, but you'll be able to see that it counts for more than one project, which is nice. See, this particular observation was added to these 3 projects. Okay, and with that, I'll wrap it up. Oops, I'll wrap it up here to close and thank Dr. Palacios Mejia for coming to our webinar series today.